In this section, we'll discuss oxidative phosphorylation. Oxidative phosphorylation is another key metabolic pathway. This is the end point in the degradation of glucose to energy. In this pathway, we take the reducing equivalents, NADH and FADH2, which were produced by glycolysis in the citric acid cycle, and convert them to energy in the form of ATP. Now, the TCA cycle, as we discussed, produces NADH in the mitochondrial matrix. And in the mitochondrial matrix, it enters oxidative phosphorylation directly. But NADH, remember, is also produced in the cytosol by glycolysis as well as some other processes. For example, ethanol metabolism, for example, as we discussed earlier. In order to generate energy, then, the cytosolic NADH has to be transferred to the mitochondria. And there are a couple of different mechanisms by which we can do that. Most tissues use what we call the glycerol phosphate shuttle. But in the heart and the muscle and the liver, there's an additional pathway called the malate aspartate shuttle, which functions to move NADH into the cell. So the glycerol phosphate shuttle basically consists of NADH in the cytosol being converted to NAD+ in a process by which dihydroxyacetone phosphate is converted to glycerol 3-phosphate. That glycerol 3-phosphate then can enter the mitochondria, in which it's converted back to dihydroxyacetone phosphate. In the process, a molecule of FAD is converted to a molecule of FADH2, and the dihydroxyacetone phosphate is transported back outside the mitochondria. And those products, the dihydroxyacetone phosphate and the glycerol 3-phosphate, are continually recycled. The net result of this is that we've taken a reducing equivalent outside of the mitochondria in the form of NADH and converted it to a reducing equivalent within the mitochondria in the form of FADH2. Now, this overcomes a basic problem in NADH metabolism, and that is the fact that there's no transporter which can move it across the mitochondrial membranes. Now, the malate aspartate shuttle involves taking NADH in the extramitochondrial compartment in the cytosol and converting it to a molecule of NAD+, transferring its reducing power to a molecule of oxaloacetate to form malate. That malate is shuttled into the mitochondria, where it's converted back to oxaloacetate, and in the process, a molecule of NAD+ is converted to NADH. This oxaloacetate is then converted to aspartate. Aspartate is shuttled back outside the mitochondria into the cytosol, and the cycle repeats itself. The net result of this is we've taken the reducing equivalents of NADH in the cytosol and transferred them to reducing equivalents in the mitochondrial matrix, where they can be consumed by oxidative phosphorylation. Now, NADH and FADH2 are reducing agents. And what we mean when we say they're reducing agents is that they have a strong tendency to reduce or to donate electrons to other compounds. And this tendency to donate electrons is called reduction potential. The process of oxidative phosphorylation harnesses this reduction potential and converts it to energy in the form of ATP. And this process occurs in the electron transport chain. The electron transport chain is a series of proteins which transport electrons donated by NADH, and this occurs in a complex of proteins called complex 1, and FADH2, which occurs in another complex called complex 2, finally donating them to oxygen to form water. The other proteins in this pathway are ubiquinone, complex 3, cytochrome C, and complex 4. Basically what happens is you have the electrons, which are present in NADH, are donated to complex 1, which donates them to coenzyme Q, which donates them to complex 3, which donates them to cytochrome C, donates them to complex 4, and finally donates them to molecular oxygen to form water. The same thing happens with FADH2, except the process starts in complex 2 to coenzyme Q, to complex 3, to cytochrome C, to complex 4, and again to molecular oxygen to form water. Each successive protein has a lower reduction potential, so the transfer of electrons is a downhill reaction, which liberates energy. You're moving from a high reduction potential to a lower reduction potential, and this produces energy. The energy in this case is used by these complexes of proteins to pump protons from the mitochondrial matrix to the inner membrane space. The net result of this is that you get an electrical and chemical gradient across the inner mitochondrial membrane. That is, the inner membrane space is now, the intermembrane space is now more positive and has a lower pH than the mitochondrial matrix does. What this means then is that there's going to be a tendency for these protons to want to flow from the intramembrane space to the mitochondrial matrix. This is another form of potential energy. Now, this potential energy generated by this electrochemical gradient is harnessed by a protein called the ATP synthase complex. The ATP synthase complex of proteins allows these protons to flow down the ionic and pH gradient into the mitochondrial matrix. And because it's flowing from an area of high concentration to low concentration, there's some energy which is produced by that transfer. That energy is harnessed by the ATP synthase complex and is used to convert ADP to ATP. Now, every NADH molecule that's oxidized results in the production of 3 ATPs. Each FADH2 molecule results in 2 ATPs. Now, there's also across the inner mitochondrial membrane an ADP-ATP antiporter that takes the newly formed ATP and transfers it into the cytosol where it can be used. And in exchange, we transport a molecule of ADP into the mitochondrial matrix where it can be recycled back to ATP. There are also in the inner mitochondrial membrane some proteins that allow protons to enter the mitochondrial matrix without passing through ATP synthase. What this does is it uncouples the electron transport from oxidative phosphorylation, and rather than using the energy to produce ATP, that energy is used to generate heat. This occurs particularly in a type of adipose tissue that we call brown fat. And brown fat is called brown because it has large numbers of mitochondria, which makes it appear brown. And it's very abundant in hibernating animals, some very cold-adapted animals, and also in human infants. The inner mitochondrial membrane in this tissue contains a natural uncoupling protein, the protein called thermogenin, which provides a passive channel for protons. So what happens in the cold is that the hypothalamus is stimulated, and via sympathetic nerves it leads to the release of norepinephrine. Norepinephrine interacts with specific receptors on the surface of brown fat cells and leads to the breakdown of triglycerides into free fatty acids. These free fatty acids then are metabolized in a process that we'll describe to you later in the mitochondria via the TCA cycle to NADH and FADH2. These go through the electron transport chain and produce this proton, electrochemical gradient, across the inner mitochondrial membrane. But rather than going through ATP synthase to form ATP, these protons pass back through the membrane via thermogenin, and this produces heat. Now, there's some other natural uncouplers. There's a protein called 2,4-dinitrophenol. This is a protein a molecule that's soluble both in lipids and in aqueous solutions, and it has a dissociable proton, as shown in the schematic below. This molecule can dissolve in the lipid bilayer of the inner membrane and can carry protons across the membrane. This uncouples electron transport from the oxidative phosphorylation and again produces heat. Now, there's some important toxins which inhibit oxidative phosphorylation, and these include things like rotenone, barbiturates, and mercurials, mercury compounds, which inhibit complex 1, antimycin, which inhibits complex 2, and then cyanide and azides and carbon monoxide, which inhibit complex 4. There's also a substance called oligomycin, which can inhibit the ATP synthase. They like to test these things on the USMLE, and so it's important that you remember which of these toxins are associated with which of the complexes in the electron transport chain. Now, oxidative phosphorylation can have defects in it leading to clinical disease, as described in the following clinical vignette. A 21-year-old man presents to an ophthalmologist complaining of worsening vision over the past few months. At first, he thought it was just time for a new eyeglass prescription, but his optometrist was concerned about the rate of deterioration and referred him to an ophthalmologist. He's even more concerned because the patient's mother and brother both developed blindness in their 20s. Vision testing revealed decreased central visual acuity on both eyes. The peripheral vision was relatively intact. The fundoscopic exam showed papilledema and microangiopathic changes of the retina. Lab tests were essentially normal except for a moderately elevated serum lactate. The disease that this patient had is called Leber's hereditary optic neuropathy, or LHON. This disease results from mutations in genes for complex 1 and complex 3 proteins of the electron transport chain. These genes are encoded on the mitochondrial genome. Consequently, they're maternally inherited. They're passed only from mothers to children because the mitochondrial genome is only passed from mothers to children, because the mitochondria are only transmitted by ova and not by sperm. For unknown reasons, the Leber's hereditary optic neuropathy is more common, though, in men than women. The age of onset is generally between 15 and 35 years of age. Now, disruption of the electron transport chain in Leber's hereditary optic neuropathy decreases ATP production. Now, this is particularly a problem in tissues that rely on oxidative phosphorylation, rely on aerobic metabolism for energy. Examples of these tissues are things like nerve tissue and heart tissue. While optic neuropathy is the most prominent symptom, patients can also have minor cardiac conduction defects and other mild neurologic defects as a consequence of decreased ATP production. In addition, NADH builds up in these cells because of decreased consumption of NADH in the electron transport chain. Consequently, this excess NADH drives lactate dehydrogenase, and you get elevated production of lactate. And so you get an elevated, mild elevation of serum lactate in these patients. So here's a summary of oxidative phosphorylation. You start with molecules of NADH, FADH2, molecular oxygen, ADP, and inorganic phosphate. At the end, you get ATP, water, NAD+, and FAD. Key enzymes to remember are complexes 1, 2, 3, and 4 of the electron transport chain, ubiquinone and cytochrome C, which are electron shuttles in the electron transport chain, and ATP synthase, which is responsible for producing ATP from ADP. The disease associated with this is Leber's hereditary optic neuropathy. So now that we've reviewed glycolysis, the TCA cycle, and oxidative phosphorylation, we can get an idea of the amount of energy that could be produced by a single molecule of glucose in the form of ATP. And we can calculate that as follows. Remember, in glycolysis, we have a net production of 2 molecules of ATP and 2 molecules of NADH. These molecules of NADH are produced in the cytosol and have to be shuttled into the mitochondria. And depending on which of those shuttles you use, either they'll be converted to a molecule of NADH, in which you'll generate 4 molecules of ATP from that NADH, or they'll be converted to 2 molecules of FADH2, which can only generate 4 molecules of ATP. So there's a difference there depending on the tissue and the type of NADH shuttle that you're using. The conversion of pyruvate to acetyl-CoA generates a molecule of NADH. So remember, we have 2 molecules of pyruvate from each molecule of glucose, and so we're going to produce 2 molecules of NADH in that portion of the cycle. And that results in the production of 6 molecules of ATP. Now, in the TCA cycle, we produce 2 GTPs, which is the equivalent of 2 ATPs. We produce 6 NADHs, because we turn the cycle twice for each molecule of glucose. And those 6 NADHs are converted in the electron transport chain to 18 molecules of ATP. We produce 2 molecules of FADH+, or FADH2, which results in the production of 4 molecules of ATP. So if you add all those things together, the total ATP production from 1 molecule of glucose is 36 to 38 molecules of ATP. This concludes the section on oxidative phosphorylation.